Hey everyone, a very good afternoon and a very happy 2022. And may all your wishes get fulfilled. You will get all the happiness and joy you desire in 2022. So it's me Abhidhi, of course, and let's begin with today's video. And in today's video we are actually going to do the body fluids and circulations, but there is a short note on it that we have already done circulation portion in the anatomy of heart. So if you want to do circulation as well, so you can definitely refer to this video of anatomy of heart, that is the episode 3 of basic biology, and it has been premiered on 12th of December 2021. Now, I hope you all remember that I showed you a meme in the Aurora or the Northern Lights video about blood only. So it says that O positive saying I can receive blood from only O, and O negative saying that I can receive blood from all blood, only my blood type. But AB positive, hearing all this conversation, says that why so discrimination? Blood is blood, everyone can accept it. But there are definitely some kind of exceptions to it. So we are going to understand this meme altogether right now. Now, your whole blood is divided into two portions, that is plasma and formed elements. So the formed elements are suspended in the liquid called as plasma. So your plasma is basically made up of water, and it is of a yellow color. So first let's discuss the plasma portion. So in plasma we have amino acids, nutrients, proteins, gases, electrolytes and nitrogenous waste. So amino acids are the simplified versions of your proteins, and then nutrients are all the nutrients such as carbs and fats and proteins as well. So why proteins is written separately? Because proteins has its own components as well. Now proteins, then we have gases such as oxygen, carbon dioxide, and electrolytes such as the hydrogen carbonate, calcium and potassium. So these are called the electrolytes. And in nitrogenous waste, in birds we have uric acid, and in humans and animals we have our urea. Now, as I have discussed that about proteins, so proteins has these sub elements, you can say, that is albumins, globulins and fibrinogen. So in the further video I'm going to discuss that how thrombin element is converting fibrinogen into fibrins. So now first let's discuss about albumins. So albumins helps the fluidity of your blood, keeps up the fluidity of your blood so that it doesn't escape from the leaks of a tissue. And globulins are definitely helping in the storage and all other things of your nutrients. Now we have done the plasma portion right now, so let's come to the formed elements. So in the formed elements we have three parts, that is leukocytes, platelets and erythrocytes. Leukocytes are known as white blood cells, or WBCs we say, and erythrocytes are known as RBCs, or the red blood cells. And leukocytes give a boost to your immune system, fight the foreign diseases, and platelets help in the coagulation of blood, and erythrocytes or RBCs carry oxygenated blood. They have hemoglobin that actually forms a compound named oxyhemoglobin when it combines with oxygen, and in that way it carries oxygen. And RBCs are devoid of nucleus, that means they don't have any nucleus. So this is all about RBCs. Now in leukocytes we have some subparts. Leukocytes, there are two subparts mainly, that is granulocytes and agranulocytes. Granulocytes means there have some granules in their cell, attached in their cells, and granulocytes are devoid of those granules attached to their cells. Now, in granulocytes, we have three basic components, that is neutrophils, eosinophils, basophils. And then let's discuss their primitive functions right now. So, neutrophils help the body to fight the infection. So, we have major kinds of infections. So, neutrophils is the one that helps us to fight those body infections. Now, eosinophils helps in fighting the parasitic infections or those inflammatory processes. So, what do I mean by inflammatory processes? So, inflammatory processes basically mean when we have some allergic responses to our environment. Now, basophils. Basophils help detect and destroy some early cancer cells. So, in our day-to-day life, we have some lifestyle habits, and they might include some bad habits as well. So, in that case, the cancer cells start like getting into our body. So, in that case, basophils helps and destroys them completely out of the body. Now, let's come to the second portion, that is agranulocytes. So, in agranulocytes, we have two portions, that is lymphocytes and monocytes. So, lymphocytes used to attack those invading viruses, and toxins. So, when a foreign like particle or foreign bacteria, virus, anything enters into our body, it tries to invade or it tries to attack our organs, tissues, and cells. So, it in lymphocytes, it actually attacks those bacteria who like try to attack our own organs, tissues, or cells. Monocytes help to fight a certain kind of infections. Very small amount of infections can be fought with monocytes, and they also help the WBCs to remove the dead or damaged cells. So, I hope you remember about basophils. We studied that it detect and destroy the cancer cells, some early cancer cells. So, monocyte help these basophils to remove the damaged cells as well. So, now group A, group B, and group AB, and group O. So, first, donor blood type. So, donor blood type of group A is A and O. Here, donor blood type means that if a person having a blood group of group A has to accept blood from someone else, the blood group of those persons should be either A or O. So, a person of group A can always receive blood from group A or group O. Similarly, in group B, a person can always receive blood from group B and group O. Whereas group AB is the universal acceptor of blood, so it can receive blood from A, group B, group AB, and group O. And group O is the universal donor, so it can give blood to group A, group B, group AB, and of course to itself as well, group O as well. So group AB is the universal acceptor and group O is the universal donor. Now, antibodies in plasma. So what are antibodies? Antibodies are a kind of protein that are produced by our immune system when a foreign substance is detected inside our body, and those substances that make our immune system respond, that produce antibodies, they are known as antigens. Like antibodies are produced only when some antigens are detected in our body. So, antibodies present in plasma in group A is anti-B, in group B it's anti-A, and in group O it is anti-A and anti-B both. But in group AB there is no antibodies in plasma. Antigens in red blood cells. So in group A it is A antigen, in group B it is B antigen, in group B it is of course, by the name we can understand it is A antigen and B antigen. But in group O, there is no antigens present in RBC. Now, coagulation of blood. So as you all remember that I have told you that from the formed elements, platelets help in the coagulation of blood. So what is the coagulation of blood? So when you get injured, so your blood starts flowing from that cut or from that injury. But it doesn't flow for all the time. It flows for some time, then it stops, the blood stops flowing. What is the reason? The reason behind this is coagulation, or in simple words we say blood clotting. Coagulation of blood is another important aspect of body fluids and circulation as mechanism to prevent excessive loss of blood. The body exhibits coagulation or clotting, and how does this coagulation or clotting happens? Let's discuss here now. Now, this is in response to an injury or trauma. I have already said that when you get injured, it like coagulation of blood prevents us from the excessive loss of blood. Now, at the injury site, a clot or coagulum is formed from a network of fibrils. Enzyme thrombin converts inactive fibrinogens to fibers. So I hope I said you that in proteins fibrinogen we are going to understand this in the coagulation of blood. So here in the enzyme thrombin converts these inactive fibrinogens to fibers, and prothrombins present in plasma from thrombins. So first prothrombins form thrombins, then those thrombin convert inactive fibrinogens to fiber. And there is another enzyme complex that is known as thrombokinase that is also required for this above reaction. And this thrombokinase is also made with a series of enzymatic reactions only. Now, again, let's just have a recap that prothrombins prepare thrombins, then the enzyme thrombin converts the inactive fibrinogens to fibers, and these fibers form get into the injury site. They form some networks or some threads. Then that is what we call the clot or coagulum, and after that the blood stops flowing, so there is no excessive flow of blood, and this is how the coagulation of blood takes place. So our body fluids has been completed, and thank you so much. It was so fun, and Abhidhi is definitely having some good vibes. So don't forget to like the video, share the video among your contacts and also with your friends, of course, and definitely subscribe to the channel for more such interesting updates and more such amazing videos. So thank you so much. Enjoy your week ahead as it is the first week of the new year, and I will meet you soon in the next week with the chemistry video, of course. Till then, bye. Have a great week ahead, and peace.